Hi. In our last video, we talked about how coloring cell specimens can be done through two processes. They were immunohistochemistry and immunofluorescence. Now, two of the most prominent types of dyes used in immunofluorescence include the DAPI dye and the Ki67 dye. And over the course of two videos, we're going to explore the two dyes. Now, both have really different purposes, but are very equally important biomarkers, specifically in the detection and treatment of cancer, so please do stay tuned. Now we're going to start with the DAPI dye here. Now its full name is 4,6-diamidino-2-phenylidol, but we won't be calling it that. We're just going to keep it short and simple as DAPI. But to understand how the DAPI dye works, we need to first understand the structure right here. Now you're probably more or less familiar with the structure. It's the DNA double helix structure. Now DNAs are in all of our body cells, with the exception of red blood cells and cornified cells, and are comprised of base pairs of adenine and thymine, and guanine and cytosine. Now, the adenine and thymine pair, normally referred to as the AT pair, comprise of two hydrogen bonds, as illustrated in the image right here, and the guanine and cytosine pair, normally called the GC pair, comprise of three hydrogen bonds, as illustrated in the image similarly. Now, to kind of make this discussion pertain to the DAPI dye, the DAPI is a fluorescent dye that binds to the AT pair of the DNA to emit a sort of like a neon bluish light. Now, the reason why this emits a neon bluish light is because when the DAPI binds to the pair, it absorbs light of around 358 nanometers of wavelength and kind of emits a blue cyanish color. So in regions where there are a lot of cells, like a high concentration of DNA and AT pairs, it kind of appears in this neon blue color. But why is staining the DNA so important? Well, it's important because it can be a very important biomarker for cancer. To see how this is, we first need to learn the concept of apoptosis. Now, apoptosis is a type of cell death in which a series of molecular steps in a cell lead to its death. The body normally uses this method in order to get rid of abnormal or unneeded cells, and is commonly referred to as programmed cell death. Now, the really important thing to note here is that during the apoptosis process, the cell symmetry is broken, and therefore the cytoplasm contracts. When the cytoplasm contracts, the cell permeability increases and it allows for more binding of the DAPI dye, so it therefore leads to a higher light intensity, like a brighter neon color. Now, the reason why this is so important is because cancer cells simply don't die. They don't undergo the apoptosis process, so they don't really emit a bright neon color when treated with DAPI. So when we try to determine the efficacy of a certain type of cancer treatment, we use this technique to see if the treatment has triggered an apoptotic cell death response, and if the cells do emit a brighter light, it signifies a successful treatment. Now, if you look at the picture I've prepared right here, it's a DAPI dye-stained human ovarian cancer cell, before and after the treatment of different dosages of piperine. And we see that there's a general increase in the light intensity of the neon blue color as the dosage of piperine increases. It's kind of like a little bit dim here, but as the dosage increases to 56 micromolars, we see that it's generally way more brighter. So since the color intensity, the light intensity increases, this means that the piperine is in fact doing a really good job at triggering an apoptotic cell response in these ovarian cancer cells. Now we're going to look at another experiment. This is a DAPI dye staining of the berberine treated HT29 colon cancer cells. And now, like, the condensation and the fragmentation of the cells, which kind of start to show at around 7 micromolars, I think, signify apoptosis of cells. We see fragmentation, condensation. Now, as the dosage increases and as we reach 28 micromolars, we see a lot more arrows, a lot more cell condensation and fragmentation. So we can see that berberine treatment is in fact effective in treating H729 colon cancer cells. Okay, thank you for watching. That's the end of this video. In the next video, we will be looking at Ki67.